Hello everyone, my name is Muhammad Hadi Ali. I'm a 6th year math student at Beirut Arab University, and today we're gonna talk about toll-like receptors and interferons. If you have any question, you could email me at my email over here. So what's the main idea over here? Toll-like receptors, they are crucial pattern recognition receptors of your innate immune system. They recognize pathogen-associated molecular patterns, which are called BAMPs, and hence trigger signaling pathways that lead to the production of interferons and other cytokines in order to launch an effective antiviral and antibacterial response. The TLRs and interferons are important to the body's immediate defense against infections. They initiate rapid, non-specific immune responses that are critical for controlling pathogens and bridging to adaptive immunity. A common mistake is confusing the diverse locations and specific PAMPs recognized by different TLRs or underestimating the broad protective and signaling roles of interferons. Before we move forward, let's discuss some key definitions. First, we have the toll-like receptors or TLRs, and as we said, they are a family of pattern recognition receptors. They are expressed on various immune cells like macrophages and dendritic cells and recognize molecular patterns associated with pathogens. The pathogen-associated molecular patterns, PAMPs, they are conserved molecular structures found on or within pathogens, but not typically on host cells. They are recognized by pattern recognition receptors like the TLRs. Interferons, they are a group of signaling proteins produced by host cells in response to viral infections and other stimuli. They literally interfere with the viral replication and activate immune responses. We have two types, type 1 interferons and type 2. The type 1, they are primarily produced by virus-infected cells and plasmacystoid dendritic cells. They induce an antiviral state in neighboring cells. Whereas type 2, they are produced by activated T cells and natural killer cells. It activates macrophages and enhances adaptive immunity. Now here's the general outline of what we are going to discuss. First, the toll-like receptors pathogen recognition process. We have the location and diversity, the PAMP recognition, and the signaling pathway activation. And then we're going to discuss the interferons from production to antiviral state to the immune cell activation, and finally the cytokine production. Now for location and diversity, the TLRs are found on various immune cells. Some of them are on the cell membrane, and others are within endosomes in the cell membrane. The TLRs located on the cell membrane, they recognize extracellular pathogens, whereas the ones located within endosomes, they recognize internalized pathogen components. Endosomes are membrane-bound compartments found within cells. They are involved in sorting and delivering internalized material. Second, we have the PAMP recognition. As we said, PAMPs are pathogen-associated molecular patterns. They are conserved molecular structures unique to microbes and not found in our host cells. Some examples include lipoproteins, lipopolysaccharides, and flagellin. These are recognized by cell membrane TLRs, meaning they are extracellular pathogens. On the other hand, we have viral double-stranded RNA, viral single-stranded RNA, and CpG DNA. These PAMPs are recognized by endosomal TLRs, meaning that they are internalized PAMPs. Finally, we have the signaling pathway activation. Upon binding of the TLRs to the specific PAMPs, they activate intracellular signaling pathways. These pathways involve adapter proteins that lead to the activation of key transcription factors such as the NF-kappa B and interferon regulatory factors. The signaling pathways are intracellular molecular cascades. They are activated by TLRs and lead to changes in gene expression and production of immune mediators. The NF-kappa B is a crucial transcription factor. It's activated by many TLR pathways and it leads to the expression of inflammatory genes and cytokines. Finally, we have the interferon regulatory factors. These are transcription factors activated by TLR pathways and they lead to the production of type 1 interferons. Now regarding the interferons, first we have production. Activation of certain TLRs, especially endosomal TLRs, leads to the production of interferons. Type 1 interferons, as we said, they are produced by virus-infected cells and plasmacystoid dendritic cells in response to viral nucleic acids, whereas type 2 interferons, they are produced by activated T cells and natural killer cells. Natural killer cells are lymphocytes of the innate immune system that can kill virus-infected cells and tumor cells. Next, we have the antiviral state. Type 1 interferons are crucial for antiviral defense. When they are released from an infected cell, they bind to receptors on neighboring uninfected cells. This way, they induce an antiviral state. This involves upregulating genes that inhibit viral replication, protein synthesis, and assembly, thereby protecting these cells from infection. Moving on, we have the immune cell activation. Interferons also act as an important signaling molecules for other immune cells. They also activate natural killer cells, enhancing their ability to kill virus-infected cells and tumor cells. They activate macrophages, increasing their phagocytic activity and antigen-presenting capabilities, and they also promote the development of specific adaptive immune responses. Last but not least, the TLR activation also leads to the production of other cytokines like TNF-alpha, interleukin-1, interleukin-6, and interleukin-12. These cytokines contribute to the inflammatory response, recruit more immune cells, and shape the subsequent adaptive immune response. As a recap, the TLRs and interferons are critical components of the innate immune system. They provide immediate, non-specific defense against a wide range of pathogens. They are the initial detectors of infection. The PAMP recognition is the ability of the TLRs to recognize conserved PAMPs, allowing the immune system to quickly identify and respond to common microbial threats without prior exposure. TLRs have diverse roles. They initiate signaling cascades that lead to the production of various immune mediators, including interferons and other cytokines that help in antiviral defense and in inflammation and immune cell recruitment. And that's the powerful toll-like receptor and interferon system. It's a fantastic example of your body's ability to quickly recognize and respond to threats. Thank you for watching and for learning about your incredible immune system.